Welcome to the Rational Alternative Weekend Edition. I'm Michael Leibowitz with my co-host, Dr. Mommy, the wonderful Megan Ribbons, and we have a great show today to talk about these three scientists I never heard of. But first, I want to talk about the origins of something real quickly, because I was just telling Megan a story, and I want to complete it. I don't want to wait till after the show. So we were talking about how trans people, I don't care if somebody wants to identify as her, him, whatever. I really don't care. It doesn't make the person the opposite sex to which they were born, but it doesn't pick my pocket, to respect their wishes. So one time when I was incarcerated, I had a guy and he was called something. I don't remember. Let's just say Johnny. And I said, well, why do you go by that and not your real name? And he said, well, that's what I prefer to be called. And I said, okay, so you think that people should be called whatever they want to be called? And he said, yeah, absolutely. I said, so you'll call me whatever I want to be called? And he said, yeah, I will. I said, okay, it's Big Daddy from now on. Of course, it didn't quite catch on the way that I hoped, but that's the those are the breaks. So we're talking about today about these three scientists whose names I'm going to let Megan say, so I'm not the one that butchers them.
Yeah, I'll throw myself in front of that bus because I will definitely butcher it as well. The first is Marcello Malpighi. The second, I think, is Jan Swammerdam. And the third, and I know this is not how it's pronounced, is Anton von Leeuwenhoek. I always try to say it like the Swedish chef. So
I think it's Leeuwenhoek. Leeuwenhoek. Mm-hmm. And see, the other thing, I would pronounce it Swammerdam. Swammerdam.
Oh, yeah, it could well be.
It's got to be Swammerdam. So can we start off
So for anyone wondering, my husband, his first language is German. So people will ask me, do you speak German? I say only to make him laugh because my pronunciation is horrible.
So I want to start with Malpighi. Is that okay with you? Or Malpighi?
I would love that.
Or Malpighi. So this guy, he was born in 1628. He was an Italian physician and biologist. He developed experimental methods to study living things. He founded the science of microscopic anatomy. And after his researches, microscopic anatomy became a prerequisite for advances in the fields of physiology, embryology, and practical medicine. All right, now elaborate.
Okay, so I think what I first want to do is pose a question to you in the audience. What if you suddenly had the means to see something you never suspected or imagined? What do you think the consequences of that would be? Not only that you could make better, more accurate observations, but observations of things you never suspected existed. What would that mean for the world going forward? What would that mean for you? So that's really how I want to bring this episode to life, right? Remember, we're setting the stage because we just talked about Vesalius and Copernicus ushering in the Copernican revolution in 1543, right? Up to this point, no one had really bothered to use their looky eyes. We were talking about the Dark Ages, right? They would debate about the number of teeth in a horse based on like what ancient text said and never bothered to walk over to the nearest horse, open its mouth, and count with numbers.
Megan, it sounded to me just now like you said nobody bothered to walk over to the nearest whores. Just so
that I cannot say.
I'm guessing they did. In the Middle Ages, everybody went to the whores, but maybe not the horses.
Yeah, probably not the horses. So go figure. Who knows? Maybe they knew how many teeth were
in a whore.
Yes. I prefer sex worker, but anyway.
Jim Valiant is here. He knows a lot about this stuff. Jim Valiant, how many teeth were in the average whore in the Middle Ages?
Wow. I
Or to
thought they
be
were
politically
debating
correct,
how many angels could dance on the head of a pin. I had no idea it was that interesting.
I can't believe that I've just gotten politically correct shamed over the word whore. It's now sex worker. Okay. But
Sorry.
proceed. It's like when prison, when we went from being inmates to incarcerated persons.
Okay. Wow. I'm so very distracted now. Were we talking about the Copernican revolution? Okay. So
You were
that
talking about
happened.
Horace's teeth.
Okay. So yeah, nobody walked over to the nearest quadruped. Just want to make that clear. A quadruped and opened its mouth and took a look inside. So it's a big change going from that, where you'd rather like debate ancient texts and like use your looky eyes, to the Copernican revolution and Vesalius's on the fabric of the human body. That, I mean, like night and day difference, right? So I mean, it would have been, I think, almost impossible for Vesalius and Copernicus to imagine the next 100 years, right? So again, imagine you've suddenly have the means to observe something you never imagined existed, right? Like, what would the world look like in 100 years, right? It's mind blowing. And so I wish, you could take Vesalius and Copernicus and just bring them to the time of Galileo, right? Who like invented a better telescope based on a vague description of the original. It's also rumored, and I don't know that this could ever be proven, that Galileo invented the first microscope. So while Galileo was discovering that Jupiter had moons, just like our planets, like again something nobody would ever have dreamed of. Um, um, Others like Malpighi, maybe 30-ish years later, and Swammerdam and Leeuwenhoek another 100 years after that, we're discovering again something that nobody had ever suspected. So I mean, I don't know how much you want to say about Malpighi, the man. He seemed like a really just sweet guy, and it's tragic that he only lived for like 40 years or something. He died young.
I really don't care about his personality. I want to know what he
Okay.
accomplished. What did he
Okay.
accomplish?
So during the time of Galileo, microscopy was I guess not in vogue. It was more of a curiosity, whereas the telescope was very much. I mean, like it's really understandable, right? You see the moons of Jupiter, right? You see mountains and craters on the moon. You're like, everyone who's not too worried about the church is all in, right? They're pretty excited about this. There's even something called like the Linnaean Society, where like a couple of Galileo's friends, groupies, you know what I mean? Like, their symbol is the lynx, the sharp-eyed lynx, because they want to see further and deeper. Of which, and I think Galileo was a member.
He had
But
groupies?
I mean, if someone accused you of being a Galileo groupie, would you deny it?
Yeah, I would. I just get the sense, when I'm with Mark sometimes has flashes of fancy where he invents things. And I'm just wondering if you've picked up the habit and you just invented the Galileo groupie.
I mean, I don't think that's too far off the mark, but they're all dead, so it's not like they could argue with me.
You got a point. Okay.
All right. So I'm wondering if I can kind of demonstrate what, like, where like the idea of the telescope and the microscope came from. So, oh my goodness, this is my ad hoc sort of experimental setup here. 1 second. Facing back. Yeah, that's what I want to do. Okay. All right. So, can you throw up my other video?
If I said no, would you listen?
No.
Okay. Here's your video.
Okay. All right. It's all rolling away. Okay, so um, I don't know if you want to unbox some of the stuff I gave you because it's kind of fun. I know it's kind of
Okay, which one do I do? Class number 4 or class number 3?
start with class number 3.
All right.
Oh no, that's the heart model. You should start with class number 4.
Okay. So I've got here 2 Ziploc bags.
Yep.
Right. This.
That's
and
for the
this.
microscope. That's for the magnifying glass. Do the magnifying glass first.
Which one's that? This one?
Yeah. So I don't know if you can see in there, but there are little 3D printed lenses with lure locks on the end, so you can screw in a syringe. So you can fill the syringe with water or something clear, and then use it to fill in the. Yep, you attach that to you remove one of the caps and loosen the other one, and then you can screw it onto the end of the syringe. And when you fill the syringe with water or alcohol or whatever you'd like, you can then fill the lens and use it as a magnifying glass.
And what is this?
Oh, that's a connector. So you can, so the way the prongs are pointing is the way it's open. So if you turn it so that all the prongs are facing sort of down and to the sides, nothing will be open to the lens. If you turn it so that the flat side is facing the
All right. So
lure lock side.
here's the problem. You've sent me more of these things than you have of these things.
That's because you're supposed to make them.
I'm supposed to make them?
Not right now, but whenever you get some plastic glue.
Right. So 500 for things that are never going to happen, Alex. But okay. So what do I do with this?
Aw, come on. You could just hold them up. So what these are meant to do, and this is what I have my students do, is make their own magnifying glasses that they can fill with all different kinds of substances that refract light differently. And here's what I mean by that. Okay, so imagine that, instead of the magnifying glasses you just have centrifuge tubes, right? And let's see. So if I shine a light on this empty one with the red cap, I'm trying to see if you can see. You can see the number 2 and the number 3 at the bottom there. If I take this one, which has water in it with the yellow cap, and I shine it next to the air one, you can see that the 2 and the 3 are bigger. And if I've got this one with oil, it's really bad oil too. I took it from my vacuum pump, as you do. It's a little dirty. You can see air, the 2 and the 3. In water, the 2 and the 3 are a little bigger. In the oil, the 2 and the 3 are even bigger.
Okay, I have a question. Are you going to tie this to Malpighi?
Absolutely.
All right.
Come on, don't you trust
that. Well.
Fair. So.
All right. Am I getting this out of here?
For the moment.
Okay.
It'll be back with fire. So the idea. So what people knew for quite a while is that if you have either a convex, like curved on both sides, kind of lens, or a concave where it collapses in on both sides, it will magnify light. And the reason it does that is because the light bends, for the same reason that I should have had this on hand. If you have a glass of water, I know, I'm just the worst. If you have a glass of water and you put a straight stick in it or a pencil or whatever, it will look bent, right? Because the way the light moves through air is not the same way it moves through water. It actually slows down. The fastest light goes is in a vacuum. I mean, that's kind of an interesting curiosity, except that there's more to it than just that it bends, right? Because if it bends like this, it means that what you have here can look bigger. So it was really Galileo, I think, who started playing around with these different geometries of lenses. And you can do this too, right? You've got curved half of lenses. So, and you've got like this 3D printed holder where you can put a lens on each side. You don't have to put the convex side out. You can put the concave side in on both sides, right? Or you can do a flat side and a concave side, a flat side and a convex side, a convex and a concave, like that's why I gave you all those little circles that you can play around with it, yes, as much as you like. So as they were playing around with this, that's where they really got good at magnifying images. So what I mean by that is that the original inventor of the telescope was able to magnify images 3 times, which is a lot more than like any kind of magnifying glass that they had at the time. Galileo upped that to 30 times. So let's see, what would be a good example? Going from this to probably that. So again, it doesn't seem like much, especially given that our modern microscopes can easily do 1,000 or 1,500 fold magnification. But again, that was enough not only to see things further away or more closely, but to see things that had never been imagined before. And so for Swammerdam, as I recall, sorry, I'm thinking of Malpighi. This is kind of ties
Do not
back.
confuse Malpighi with Swammerdam.
I'm sorry.
I
They
will not
two
have
different
it.
people.
I will not have it.
That is fair. I am sufficiently contrite. Anyway.
Okay.
So Malpighi, among other things, I just think this is so cool. He dissected a living frog. I mean, okay, it's bad for the frog, but he could see the lungs expanding. And he could even, with a little bit of microscopy, look very closely at the little air sacs in the lungs and see capillaries. Now, this was the mystery that Harvey had to live and die with a couple of weeks ago, right? He, along with Cecilius, was so in love with the human form, and he figured out that blood circulates, and he had a lot of ways of proving it, but he could not for the life of him figure out how you go from arteries to veins, right? He knew that arteries is where the blood was going directly from the heart straight to the arteries, right? Because when you cut it, it squirts, as you'll see with your heart model. Whereas like coming back to the heart, right, in the veins, if you cut the vein, right, the blood oozes, right? There's a reduction in pressure. How did those two things connect? It was Malpighi who figured out the answer. He could see the tiny microscopic capillaries. And because the frog was still alive, you actually need, like living tissue in order to see this. He could see the blood with the help of microscopy going through the capillaries around the frog's lungs into other capillaries that would then sort of drain into the veins of the frog. So he literally closed the loop on the circulatory system.
All right. So now Jean Swammerdam, he's baptized February 12th, 1637. So I don't know what day he was born, but I know when he was baptized. He's a Dutch naturalist, and he was considered the most accurate of the classical microscopists. He's also the first to observe and describe red blood cells. He completed a general history of insects, which was a major work at the time, and the Bible of nature. He corrected Malpighi on the insect brain and nervous system, and he opposed William Harvey for being inconsistent with his interpretation of insect metamorphosis. All right, Megan, now that I've read from my phone the description of Swammerdam, take it away.
My impression of him, and again, I'm no historian, so take this for what it's worth, is that Swammerdam was kind of a firebrand. He did not mind pissing people off. So why was like a lot of the work I'm familiar with regard to Swammerdam is with regard to insects. So why is this important? Like, what is the cash value of this today? Well, again, he saw something that nobody ever suspected. Back then, people thought that caterpillars, pupa, and butterflies were completely different species. I mean, imagine how, like when you look at, a baby to a human, you can kind of see what's coming, right? There's not new limbs or new organs, right? It's all just kind of in the same general shape. So it must have been a revelation to discover that in one life cycle, you've got a thing with no limbs that just kind of wiggles around to a thing with a bunch of limbs and also like wings. What? Yeah, like again, it was something that nobody suspected, and again, must have been a revelation. So in part, it's just makes you appreciate more just how weird and wonderful living things are, that you could have these bizarre, unpredictable metamorphoses. But that will also become very important to Francesco Redi, who I think we'll probably meet next week. Also another like Galileo kind of groupie, who did the first controlled experiment in biology. So keep that in mind, this idea that, here's Swammerdam showing at about the same time Redi was doing his work that, maggots and flies are not completely separate species, as would be totally reasonable to assume. No, they're just child and adult of the same species. Okay. So the last one is Leeuwenhoek, who I'm a little bit in love with.
You're in love with Leeuwenhoek?
Oh God, he's adorable.
Okay. So Antoine van Leeuwenhoek. He's from the Netherlands. He was born in 1632, died in 1723. Also a Dutch microscopist. He was the first to observe bacteria and protozoa, and his researches on lower animals refuted the doctrine of spontaneous generation, and his observations helped lay the foundation for the sciences of bacteriology and protozoology. So before you go sick, I got a couple questions for you. First question is this: what are bacteria and protozoa?
Well, at the time, they didn't know what that was. And how do I put this? It was another thing that they never suspected existed, right? So I don't know. At least for me, it's hard to imagine a world where I wouldn't have suspected bacteria existed. That was, I was about 11 or 12 when that, like that notion was presented to me, which is some time ago. So it's hard to remember before then what I kind of thought of the world. But put yourself in the shoes of people back in the 1600s, right? They'd seen vicious plagues destroy like 50% of certain town populations. Like that was pretty normal. It wasn't great by any means. But that's what they knew, right? They couldn't see why this was happening. They actually thought it was bad air. Malaria means bad air. And imagine how terrifying that would have been, right? Like normally when you get hit, if someone throws a grain of sand at you, it doesn't hurt nearly as much as if they throw a boulder at you, right? Normally bigger things cause more damage. So the idea that something even smaller than a grain of sand could kill you was really bizarre. And something I don't think anyone really considered, except, something in the air, right? So Leeuwenhoek, like at this point, the microscopists and people with telescopes are playing around with two lenses. And that's why I gave you two syringes, is that you could, if you wanted to, you could make your lenses and you can put them in order and see if you can. Let me see. Do you want to throw my experimental camera back on? See if I can do this. This is so. I need like 5 hands. I have 2. Okay.
Sure. And just so everybody
Okay.
knows, this is the Rational Alternative Weekend edition. On this show, we discuss a lot of science stuff because, well, Megan is a scientist. During the week on this channel, we have the Capitalist Corner with Mark Pellegrino and myself, where we discuss politics from a pro-reason, pro-capitalist perspective. We also have the Michael Leibowitz Show and membership-only content. Please subscribe to the show. It'd be greatly appreciated. And also, remember, super chats and super stickers are the bomb. All right,
Yeah.
Megan, go ahead.
Agreed. So I don't know if you can see this, but I'm putting the water tube in front of the oil tube because both of them will magnify. So as I move them, and I'm really struggling with my camera work here, as I move them backward and forward, there's probably a sweet spot there where you can actually get a clear image that is higher magnification than either one alone. So that's one of my suggestions for things to play with, because you don't have enough to do. I say this to the hardest working person in show business, so yeah, take that for its worth.
Okay, now focus. What is bacteria and what is protozoa?
So they are both kinds of microscopic organisms.
Okay.
If you were to look under the microscope, you need a pretty high magnification. And that's where I was going with that. While most microscopists were playing around with 2 lenses to get even better magnification, Leeuwenhoek kind of took it in a different direction. He was using one very small lens, and he made like 200 of his own microscopes because he got really good at making single lens, very high magnification microscopes. And that's another thing you are able to do yourself. So what are bacteria and protozoa? Well, bacteria are often found as single-celled organisms. So if you see them under your microscope, you might see a sphere or a kind of rod. Let's see. You won't see much inside them. They'll appear pretty transparent, like little balls, right? But you might see some of them wiggling. If you're seeing protozoa, they'll usually be a little bit bigger, spheres or rods. They may be colorful. You'll probably see more distinct structures. And if you use special dye, you might even see structures within structures, because protozoa actually have, you remember we were talking a few weeks ago about the difference between eukaryotic and prokaryotic cells. Bacteria are prokaryotic. They don't have that special structure near the middle that's containing something very, very important. But the protozoa do. I'm not going to tell you what that very, very important thing is yet, because we don't believe in spoilers in this family. All right, can I turn this on? All right, so now, how did Anton von Leeuwenhoek, or better question, how can you make your own microscope?
But I want to know this first. Hold on. Yeah, I'm trying to learn here, Megan. You're jumping the gun. Are protozoa, like bacteria can harm you? Can protozoa harm you?
Yeah, I mean, there are, I think, many fewer pathological protozoa than there are bacteria, but there are also fewer species of protozoa.
Okay. And Jim wants to know, when did they come to understand that microbes are living organisms just like bigger plants and animals? That might be a tough question. I don't know.
I mean, Anton van Leeuwenhoek was the first person to discover microscopic organisms, period. And he spent decades just like exhaustively writing down what he observed. And hopefully we'll be able to see a little of what he saw today. I actually need one more thing, but fire. So we're going to do fire.
Okay,
Okay,
one
let's do
more
it.
question. Hold on. You're
All
so
right.
eager. You're so excited, like a kid in a
Of course I
candy store.
am.
But for those of us who are a little slower, I want to know this doctrine that he showed to be false of spontaneous generation. What's that?
So, again, if you've never known that maggots become flies and that there are invisible living things in rainwater, which is another thing that Leeuwenhoek discovered, you might think that pond scum, right? Like you've got a fresh trough of water for your horses, right? Um, and a few days later, there's a scum that forms on it, right? The scum looks alive, you know what I mean? It looks like kind of a amorphous plant. Where did it come from? I mean, you'd have pretty good reason to think that it just spontaneously generated, right? Or you have a dirty basement and all of a sudden you have mice in it. And you're like, I don't know how the heck they got in. They probably just formed that way. You know what I mean? They just probably kind of form and fill. That was a perfectly reasonable presumption that I think most people had. And yet there were people who were saying, I don't think that's quite right. So it actually took several centuries to completely disprove um, the idea of spontaneous generation, that life just kind of appears where conditions are favorable. And that story actually ends with Pasteur, with Louis Pasteur. So we will definitely get there. Ready is, for next week, is also part of that story.
So I remember when I was a little kid, Megan, right? A friend of mine, there was a bucket left outside and it rained. Heavy rain, the bucket filled up, and nobody ever bothered to dump this bucket out. And after a while, and I don't remember how long it was, I was about 8, there were things swimming in that water, living things. And I remember being bewildered, like, where the hell did these living things come from? Where the hell did these living things come from that were swimming in that water?
Um, some of them probably from the rain. You know what I mean? So this goes a little beyond Leeuwenhoek, but so Leeuwenhoek actually took fresh rainwater, just collected it, put it straight under his microscope, and saw things moving around in there under his microscope that he couldn't see with his naked eye. He's like, I bet if there's a whole lot more of them, you'll be able to see them without a microscope. And he was absolutely right. But it turns out that there are bioaerosols. And I really want to build a high altitude weather balloon to sample the upper atmosphere, to the point they can get so high that you can actually see where the atmosphere goes from blue to black. And I really want to get samples of bacteria, because there are bacteria up that high in the atmosphere, right? Dust gets kicked around, and some air goes up that high, and eventually it can come down, especially when water nucleates around bits of dust and stuff like that and falls as rain. Yeah, it's pretty incredible.
Okay, now you can just launch about Leeuwenhoek.
Okay, so one of the things you can do in my class, especially if you buy our class 4 materials, is make your own microscope. So what Leeuwenhoek did was make very, very tiny lenses. Now, why does that work? Why was it so much better than 2 lenses? Well, it has to do with curvature, right? If you take a clear marble, right, it will bend the light. The smaller the glass marble is, the more it bends the light, because it's still a sphere, but it has to go 360 degrees in a shorter distance, right? Because it's smaller. So there's more curvature. It has to bend light that goes through it more. So a good example is if you have a sort of spherical bowl of water, right? It will bend the light a little bit. Your stick will bend a little bit. If you put a glass marble in front of that same stick, it bends much more. So what Leeuwenhoek did is have very, very tiny microscopes. This is not original to me. There's this professor named Keeling, I want to say. I've got links in my YouTube channel that figured out how to make your own Leeuwenhoek microscope. So what you do is you take this little glass. I mean, you can take any piece of glass if you really wanted to. But what's easiest to do is take a little piece of a glass capillary, like what they do for if you're getting your blood tested to donate plasma or something. So it's a tiny little glass
What is a
cylinder.
glass capillary? I thought capillaries were in blood. What is a glass capillary?
Oh, it's just a tube. It's a tube
Okay.
of
All right.
glass.
All
It's
right.
really, really
Okay.
tiny. So
Okay.
let's see. And you take a good flame. I tried with a candle flame, but it wasn't very hot and it didn't really work very well. So I don't know if you can see this, but this nice hot blue, I assume it's butane flame, could be propane, is making a little dot of glass at the end. Now, if it looks like a lollipop instead of a teardrop, that's a really good sign. But you can see it's a very, very tiny bit of glass. And I'm trying to get it just right in front of the camera so that you can see me behind it, except that, like, yeah, my arm is wiggling. Because my image will appear upside down. I can show you a larger version of this. So when I first started, I didn't save any of my magnifying glasses. I sent them all to you, which was stupid. But if I can put this in front of the camera.
That's the same thing with the human eye, right? That the images in the human eye appear upside down, don't they?
Yep,
Is it for the same reason as with that?
Yes.
Okay.
Yes. So you, yeah, it's really not working. But so when I first started prototyping the magnifying glasses you can build, this is a much earlier version. But I used to chase Gracie around at like school. There'd be like school picnics and stuff like that, and she'd be so annoyed with me. I'd be like, turn that frown upside down. I don't know if you can see that, like I'm dripping on my keyboard, but like because of the way the light bends, the image flips upside down. So when I fill the double convex lens with water, it flips my smile upside down okay. Anyway, sorry, that was such a bad joke.
That was a joke?
Come on.
What was the joke?
Like humor's because you like turn the frown upside down. You hold the thing in front of her frown, and like the image turns upside down because
you just said it turned your smile. You said it turned your smile upside down.
Well, I was looking at her frown, so you know what? My comedic gold is lost on you. I'm so annoyed with you right now. I'm not that annoyed with you.
Science humor, great. So the human eye, I'm interested. I'm going to take you off on a little tangent here because I'm interested about this. So it's the way that the lens of the human eye is bent. That's why images appear upside down.
Mm-hmm. I mean, you can tell that your eyeball is pretty much spherical, right? That looks a lot like the glass marble we were talking about, or the Leeuwenhoek microscope.
Yeah. Yes, convex, the human eye, correct? It'd be like if you took two convex things and put them together.
Yeah. Mm-hmm.
Okay. So then what happens after, once the image is upside down in the eye, the optic nerve flips it back up before it sends it to the brain? How does that work? Do you know?
No, I don't think it does, because the optic nerve just kind of goes to the back where your visual cortex is. I think your brain has to interpret it right side up. Which, I mean, if you ever want to experiment with that, you can put on some
Barry White and just get nuts.
I wish I'd thought of that, but no, what I was actually thinking of was you can get like some silly glasses or something that distorts the image. And eventually, so if you have like a bullseye where you're like throwing a ball, right? If you have aim, which I don't, but if you do, right? Then you put on the glasses, right? You'll throw the ball and it won't go anywhere near the target. Eventually, you'll get it to hit the target. And when you take the glasses off, you won't be able to hit the target right away, right? Because your brain has adjusted to the distorted image. So it's, I think, and I'm no neuroscientist, so take this for what it's worth. I think that's what's happening with your eyeball images.
I think that objectivists need to leave the jokes to Jim Valiant and me. That's what I think.
Well,
Jim, what do you think? Should they just leave the jokes to
wrong.
us?
Okay, so let's see if I can get this just right. All right. So I'm putting the Leeuwenhoek microscope I just made in front of everyone.
Okay, you're just blocking everybody's view. Nobody can see anything now.
Take a minute, take a breath. And I've got, hang on, you put up my other thing here. So this is water from my dog's outdoor water bowl. You know, it's outside, so it gets pretty scuzzy. What I'm going to do is I'm going to put it up to the microscope, get some backlighting there, and can I get it to focus? That's always the tricky part here.
I think you're starting to stress everybody out now. First, you come with a bad joke, and now you're blocking the camera so no one can see what the hell's going on.
Yeah, sometimes it works better than others. Anyway,
I don't think Leeuwenhoek had this problem.
well, no, he probably didn't have a problem with his webcam not working. It usually works better with my webcam than it does with my phone. But when I get it to focus just right, I wonder if I can keep futzing with it while you talk. You can actually see the
Oh, look at
water.
that. Everything just got weird now.
Let's see. I don't know. It just I love I'm trying to like focus the pond water. That's the tricky part. Anyway.
It's freaking me out. Okay, Megan, tie it all to Leeuwenhoek.
So yeah, in my class, you can make your very own microscope, and it's probably just that the backing isn't working very well. And you actually can get it to focus and see little itty bitty creatures in pond water, or if you want to look at the wings of a fly, which we will definitely do later on, so it might be helpful. You'll definitely want that magnifying glass if you want to look at flies, because we are going to do that at some point. But in any case, so Anton van Leeuwenhoek made these teeny tiny lenses, and because they were spherical and so small, they bent the light really, really well. He got amazing magnification to allow him to see microbes, things that you cannot see without really good microscopy, things like bacteria and protozoa and archaea.
Christopher Smith, just so you know, icy dead people are definitely solid material. Just to keep it real.
Thank you, Christopher Schmidt. I like to think that somebody appreciates my humor. But in any case, yeah, did that answer your question?
I have no idea. I'm kind of lost. This stuff all kind of confuses me, which is why I just ask my own questions and learn what it is that I want to learn. But what I'm very interested in is, why did you pick these three, or why did you group these three scientists together? Malpighi, Swammerdam, and Leeuwenhoek. And where did you go? Get back over here.
I'm trying to get it to focus. My webcam's being a total pill. This normally works, and I've done this demonstration on air in real time before, and it worked way better. So I'm really disappointed with myself that it's not working. I don't know if you can kind of see a little bit that like I'm moving my camera. Anyway, so first of all, they were contemporaries, although Leeuwenhoek lived a lot longer. Secondly, again, they were taking this new technology and making brand new observations. And I can't tell you, I don't know, this is definitely coming from personal anecdote, but these days, very few people think a lot about observation at all. They treat it a lot like it's dispensable, which is really tragic. It's sort of
just learn math, right? It's all math.
Yeah, essentially. So, like I told you a couple of weeks ago that I was giving a talk to this group where they were trying to manufacture organs for, because there's a big difference between the number of people who need transplants and the number of transplantable organs. So they're trying to bridge that gap. It's really important. and they'd follow the same procedure and they'd get different results. It was like everything was the same except the output. And they're like, what are we missing here? I'm like, more observations. There's no getting around that. If none of your observations are correlating, probably not related to the cause. You need new observations. That went over like a sinking ship. I'm sorry, I couldn't come up with a better analogy. It did not go over well.
That doesn't make any sense. This is another Pellegrinoism. Do you guys like watch the show to pick up hit tips from him? It went over like a sinking ship. You mean a lead balloon?
Yeah, thank you, like a lead balloon. That's even better. Sorry, I haven't heard that phrase in decades.
So you figure you just make up your own?
I mean, that was the best I could do. Sorry.
Well, I mean, you could say that went over like a whore in church. I mean, there's a lot of different ways you can say things like that, but like a sinking.
Church, so how would I know?
Well, you could use your rational mind. Churches frown upon whores or whore behavior, so they're probably not going to be very whore welcoming. Although Jesus,
way too much credit for my theological knowledge. It is non-existent.
Oh, well, Jesus liked the horse. But either way, back to why Leeuwenhoek and the other two, you group them together.
So, I mean, I wish I knew how to say how important what they did for us was. Not just discovering weird metamorphoses in insects, and not just providing us data that supported this idea of there's no such thing as spontaneous generation, which ultimately led to the cell theory, right? Which was essential to modern medicine, right? It's all connected. And it's more than that. They made observation important after 1,000 years of pretty much no observation. And man, did they hit pay dirt, right? Like, to discover things that nobody could have even suspected.
Why did they hit pay dirt? Why did they hit pay dirt, Megan?
Because again, they discovered that yes, the circulation of the blood is a real thing, even though we can't see part of it. Yes, maggots don't, like flies don't come from rotten meat. They come from other creatures, just like people don't come from rotten meat. They come from other people. And yes, there are small invisible organisms that can cause disease. It's not bad air. There is a real cause that we can identify and deal with.
Megan, I'm going to save the day. You ready for me to save the day on your grouping of these three?
I'm ready for you to mansplain.
I'm going to mansplain. So yes, it is putting the Christ before the whores, Jim. You were right. And that's why I said Jim is allowed to joke, because his jokes are funny. Fundamentally, in my view, Megan, and you can correct me if I'm wrong, what these three had in common is they were able to make new discoveries because of new technology. They were able to see.
Is that what I just said?
No, they were able to see. They were able to see what previous generations couldn't. You said they saw it, but you didn't say why. And the reason they did is because of new technology. New technology that cannot just spontaneously come into being. It has to be created, invented, discovered. Knowledge builds on knowledge. So Aristotle would not have been able to make these observations, for instance, because he didn't have the technology. But now they did have the technology, and they were able to come to new discoveries. And with these three especially, it seems that all three relied on the microscope.
Yes, they do. Sorry, I thought that was just part of the subtext, so maybe I was being too subtle. Is a thing that I never do.
Okay, so Megan wants to take credit for my conceptual grouping of the fundamental characteristics of why these three are put together. We're not going to allow Megan to get away with that. She's a brilliant biologist, but I am here for something.
Thank you. I guess. So I don't know, like, I see this every day in my job, right? Like, I get to work with some amazing technologies that allow for amazing observations. So as an example, there's something called mass photometry. It's where there's a laser and you put a glass slide over it and take a tiny droplet of water, and the the stuff inside the droplet of water, whether it's a virus, right? So something easily 1,000 times smaller than a bacterium, or even an antibody, which is 1,000 times smaller than a virus, you can see them stick to the slide. And then the computer will kind of, ignore that. Kind of, it's like it's wiping the slate clean. But you see these dark spots, and the darker and bigger the spot is directly correlated with how big it is in terms of its molecular size, right? So the deeper the shadow, the bigger the molecule. And you can see molecules and viruses dancing on this slide. And every time I look at this instrument, I fall in love with human beings all over again, because somebody invented this. And like the person who did talks about his kid and how like they were looking at Where's Waldo and how do you see Where's Waldo? Well, it's really easy if you can subtract away everything that isn't Waldo. You know what I mean? That's what he does with his instrument. It's the coolest thing. So it's tragic to me that the zeitgeist these days is that observations just aren't that important. We have all this new technology to have brand new insights into the living world. And we don't think that it's important, that we couldn't learn something new and unexpected. So yeah, I just want to reemphasize before we get to the experimental part of this show that start with observation. Don't skip that step. If you haven't gotten your herbarium samples, be a Leeuwenhoek, be a Linnaeus. Go do it.
So Katie says, sorry, Mike, I choose Mark in Chicago over you tomorrow. That's okay, Katie, because you did give $4.99 in the super chat. And for that, you have bought yourself, what's the word I'm looking for? A dispensation. You are forgiven. Resilient Batist, he's made this comment, or she, sorry, my sexism showing through. Observation is important, but we have this nasty habit of deleting the observer. Previously had said there's no such thing as a neutral observer. This, I think, can be taken in two ways, Megan. I don't know from which perspective the person's coming from, but either Einstein's idea that everything's relative to the observer, or the quantum idea that nothing can be observed without the observer affecting that which is observed. I don't know which perspective the person's coming from, but if we factor in the observer, I don't think that takes away from the truth or importance of observations, not that the person is saying that. What do you think, Megan, about this?
I think this person makes a good point. And again, I don't know what exactly they're getting at. So if they want to follow up, please do. But I mean, Malpighi, Swammerdam, and Leeuwenhoek were all very different human beings. And they all contributed something irreplaceable and revolutionary, right? So yeah, I'm glad we know about these observers, right? I love introducing different scientific heroes, right? Because we can relate to at least one of them, right? Like, you and I struggle with mental health, right? Which is why I'm so grateful that there's a Marie Curie and a Gregor Mendel who also struggled and still contributed something irreplaceable to human knowledge and well-being, right?
Did you just out my mental health struggles?
Well, long after you did. Was I not?
No, I'm teasing you. Yes, no, I'm mentally screwed. But, it's interesting that you say that.
But you can still be a Curie or a Mendel, right? Like, yeah, it sucks, but it you shouldn't think it's
You ever hear of Gary Vaynerchuk?
Stop you.
Do you know who Gary Vaynerchuk is?
Mm-mm.
Okay, I think he's a billionaire, but I might be wrong. I might just be multi-millionaire. He also does motivational speaking, and he does a lot of swearing. Right? So I saw a video of him recently, and he says, people ask me, "Do you have ADD?" And he says, "How the fuck do I know? And I don't really give a fuck. Like, okay, I have ADD. So fucking what? I'm going to go on and do what I got to do anyway." And I think that's the same with the mental health stuff. I don't know how we came around to that, but nonetheless, I think it's a good point that we all need to learn.
Well, thank you. Yeah, my point is that, scientists aren't monoliths, and I don't think the observer should disappear because they're actually people too. I often say this, like, yes, I may be a scientist, but I'm also a person. You know what I mean? And I think those personalities are a great part of the story. It means that anybody can contribute, anybody can observe, can use their looky eyes, and make discoveries that advance human flourishing. Now, what's important about these observations wasn't just that they used their looky eyes. It's that they observed in a way that could lead to correlation, right? And that's the difference between just casually noticing every time you're stuck at a red light versus every time you get to go through. It's when you actually start counting and noticing the time of day that you can make the correlations, right? That it's not just about noticing. It's about noticing in a really rigorous and even better, if you can, mathematical way, because math will tell you if there's a correlation. It'll tell you if that correlation is linear or exponential or logarithmic. And each one of those kinds of relationships tells you something very important about what's going on. So sorry, I'm about to, like, yesterday I spent an extra hour at work just talking about math. So if I get on a tangent on math, we could be here for a while. And I know we've got roughly 3 minutes.
No tangents on math. Next week, who'd you say we're going to discuss? The ever ready? What was Freddie? Who?
Francesco Reddy.
Francesco Reddy. We're going
So,
to discuss next week.
and I'm sorry to put you on the spot. How would you feel about receiving some fruit flies?
I don't care.
Okay. How would you feel about working with fruit flies?
I can work. Are they living or dead?
Alive.
I hate flies. Hate them.
All right.
If these things, listen, are they going to get loose? If they're going to get loose, I don't want anything to do with them.
I can help you knock them out. That's routine.
Okay. Yes, because
and I promise there will not be escapees. No. So maybe I should do something else for you.
No escapees. As long as there's no escapees. Jim says, bless the obsessed. Bless the obsessed. Yes, the obsessed shall inherit the earth. All right, what a great show. Thank you, everybody. Again, if you joined from X, check us out on the Rational Alternative YouTube channel. Please subscribe. We're almost at 1,000. We're growing. We're getting there. Megan, what the hell was I going to say? Tell them where they can find you.
All right.
Besides here, of course.
Of course. So also subscribe to my channel. I'm trying to monetize, and I'm at a whopping 134 subscribers. So subscribe, make a sock account, subscribe again, share it with your friends, share it with your enemies, anyone who has a kid or knows of a kid or has seen a kid, subscribe all the way. And that is at at MARIBBENS, Megan Ribbons. And also shoot us a tip on Patreon so I can keep sending Mike things that can bewilder him, because these FedEx shipments are not cheap for whatever reason. That's at patreon.com/DRMOMMY.
Awesome. Thank you, Megan. Thank you, audience, for joining us for another great discussion. Hopefully, I'll see you tomorrow with my members only show. If not, hopefully, I'll see you Monday with Mark Pellegrino. And if not, hopefully, we'll see you next week. Either way, have a great rest of your day and weekend.